السلام عليكم أعزائي طلبة المرحلة الثالثة في كلية الطب جامعة الموصل. أنا الدكتور وليد خالد سعدون التدريسي في فرع الأحياء المجرية في كلية الطب، واليوم إن شاء الله سأواصل إعطاء مختبرات الجراثيم العملي للمرحلة الثالثة لهذا العام. Our lab today include two important subjects. First of all is isolation of microorganisms from the environment, and second is genus Staphylococcus. What are the objectives of this lab? At the end of this lab, you will be able to isolate the microorganism from the environment. Also, you will describe a colony morphology on a garden, and also you will identify the genus Staphylococcus based on their microscopical and cultural characteristics. So let's start with isolation of microorganisms from the environment. The microorganisms are found throughout the environment. They are found everywhere. Found in the tables, on the ground, in the air. And the best method to isolate the microorganisms from the environment is what is called as a pure culture. So what is the meaning of pure culture? A pure culture means to isolate a single kind of microorganisms and allow it to grow in a protected environment. What are the best methods to isolate the microorganism in a pure culture? This method is called as citric plate method. It is the best method to isolate a pure culture from mixed population, like from urine or sputum or pus, or even from a table. Now the table contains a mixture of microorganisms, and in order to take a pure culture from this mixture of microorganisms, make a citric plate method. So what is the technique of the citric plate method? Citric plate method is, as I said, is the best method to isolate a pure culture of microorganisms. Now, for example, I want to take a pure culture from the table. So what are the steps to make the citric plate method? First of all, I will touch the table by a cotton, and I will work in this way. I will make an inoculum, and from the inoculum, I will sterilize the bacterial loop and make an area A by pulling 3 lines. Then again sterilize the loop in benzene flame, and from the distal part of area A, I will pull 3 lines to make an area B. Again sterilize the loop and make an area C from distal part of area B, and then again for the area D. And this is the explanation. Now, after touching the table, I will make an inoculum on a clean nutrient agar. Then I will take a bacteriology loop and I will sterilize it by the flame, cool it, and from the inoculum I will draw 3 lines. This is an area A. Again I will sterilize the loop, and from the distal lines I will make an area B, sterilize the loop, cool it, and I take an area C. Again from the distal end of the lines of area B, again sterilize the loop, cool it in an empty area, and make the area D from the distal part of area C. By this way, we take an areas A, B, C, and D from the inoculum, and in this way we will make a filtration of the colonies of bacteria. Now, here if we have a mixture in the inoculum, if we have a mixture of bacteria, then it will be filtrated, and in areas A, B, C, and more in area D, in order to take a single separated colonies in area D. After that, we will incubate the agar in the incubator at 37 degrees centigrade for 24 hours. After incubation, after 24 hours, we will take the agar to see a mixture of bacteria, a mixture of colonies on the agar. Because the table contains mixture of bacteria, and each bacteria will produce a specific type of colony. So we may take a plate like this. As you see here, there are different types of colonies because it contains different bacteria, and each bacteria produces a specific morphology of colony. Now this culture is not a pure culture because it contains a mixture of bacteria. In order to obtain a pure culture, we will make what is called as subculturing, in which we will take one of the colonies that we need to study, like this. I take a part of this colony, and again I will inoculate it on a clean nutrient agar, make a inoculum, and then make areas A, B, C, and D as mentioned previously, and incubate it for 24 hours at 37 degrees centigrade. The culture produced in this procedure will be a pure culture because it will contain only one type of colony, like this culture. Now this culture is a pure culture because it contains only one type of the colony. It's prepared from a single colony that is needed for the study. Now, after appearing of colonies on the agar, we have what is called as cultural characteristics. Cultural characteristics includes 2 points. First of all, the growth requirements, and second is the colony morphology. The growth requirement of the bacteria include the best environment that is suitable for the bacteria to grow, like the best temperature, the best pH, or the best NaCl concentration, or anything else. This is about the growth requirement. The second point is the colony morphology. Each bacteria produces a specific colony, and in order to describe a colony, the colony morphology, we have to include these 10 points in our description, according to the shape of the colony and the colony that we see by the naked eye. Now the first point is the size of the colony. It is usually measured in millimeter, and there are specific tools that are used for measuring the size of the colony. And for you can say it is large, medium, or small size colony. Second is the shape. There are rhizoid, circular, filamentous, or other shapes of the colony. Also, you have to include this in description and colony morphology. We have the elevation. Elevation, we have either a flat colony, or slightly raised colony, or convex colony, or umbunated colony. And sometimes it is better to see the elevation of the colony by looking at it from the side. The fourth point is the margin of the colony, whether it is entire, or it is lobulated, or filamentous, or other types of margins. Also the consistency. The colonies may be dry or mucoid. Now for the mucoid colonies, we have a test called a string test, in which we will touch the colony by the bacterial loop and elevate it. If the colony is a mucoid, then a thread will be transferred from the colony to the bacterial loop. It will be elevated with the bacterial loop. This indicates that this colony is a mucoid, like in Klebsiella pneumoniae. Also, we have to look to the surface texture, whether it is smooth or rough. The color and pigmentation. The color is the color produced by the colony. As you see here, some colonies will produce yellow color, or red, or orange, or different colors, white color. So every colony will produce a specific color. This is the color. While the pigmentation is the color that is diffused to the media. As you see here, this is called as pigmentation. It is diffused, the color, the yellow color is diffused throughout the media. So the color is what is produced by the colony itself, while the pigmentation is the color that is diffused to the media. Also, we have the optical density, whether it is translucent or opaque. The changes on the medium. For example, blood agar. Now, some colonies will produce hemolysis on blood agar. And we have three types of hemolysis. Either alpha hemolysis, in which there will be greenish discoloration of the colonies, or beta hemolysis. Beta hemolysis, here the colony will produce a clear zone around the colony. So as you see here, there are two types of colonies in this plate. The first one, the separated colony, will produce a clear zone around the colony. This means that this bacteria is beta hemolytic. While the other colonies, there is no clear zone around the colonies. This means that this bacteria is non-hemolytic bacteria. The last point regarding the colony morphology is the odor. Now, some bacteria produce a characteristic odor, some bacteria produce a sweety odor, and others produce fishy odor. And you will see this in the next laboratories. This is all about the isolation of bacteria, of microorganisms from the environment. We will pass to the second part of our lab today, which is the genus Staphylococcus. So we will start with the systemic bacteriology, and the first bacteria is genus Staphylococcus. We will discuss in this part the classification of Staphylococci, the microscopical appearance, the biochemical reactions, and the cultural characteristics of genus Staphylococcus. First of all, regarding the classification, they belong to the family Staphylococci, genus Staphylococcus, and this genus contains more than 40 species. Only one species is pathogenic, which is Staphylococcus aureus, while the other species are non-pathogenic. And some of them may cause infection when the immunity decreases. Like the most important two non-pathogenic staphylococci are Staphylococcus epidermidis and Staphylococcus saprophyticus. Also, according to the coagulase production, we can classify staphylococci into two types: coagulase producing staphylococci, or coagulase positive staphylococci, and coagulase negative staphylococci. This is the term. You can phrase this term, the coagulase negative staphylococci. Sometimes we name them as CONS, coagulase negative staphylococci. It includes two types of staphylococci, which are Staphylococcus epidermidis and Staphylococcus saprophyticus, while Staphylococcus aureus is coagulase positive staphylococci. This is about the first point, which is the classification of genus Staphylococcus. Now, regarding the second point, the microscopical appearance of Staphylococci, they are gram-positive cocci. It means they are violet in color and circular in shape, and they arrange in single, pairs, tetrads, or more commonly in irregular grape-like clusters. And each cell being approximately 1 micrometer in diameter, spherical, non-motile, non-spore forming, and usually non-capsulated. The most important point here is that all staphylococci will give us the same microscopical appearance. So, by microscopical appearance, we cannot differentiate between species of staphylococci. All of them are gram-positive cocci arranged in irregular clusters. The third point is the biochemical reactions. We have two important biochemical reactions, which are the catalase test and the coagulase test. Catalase test is a key test of differentiation between staphylococci and streptococci, since both of them are gram-positive cocci. So how can we differentiate between them? We can differentiate between them by catalase tests. Staphylococci are catalase positive, while streptococci are catalase negative. Now, how can we make the catalase test? First of all, we will take the H2O2 which is hydrogen peroxide, and this is the Staphylococcus, genus Staphylococcus. Now we put 1 drop of H2O2 on the suspected colonies. As you see here, there's formation of bubbles. Now, there are formation of bubbles. This means that this is the Staphylococcus, genus Staphylococcus. Now, why these bubbles are produced in genus Staphylococci? Because they contain the catalase enzyme, and catalase enzyme will liberate free oxygen from the H2O2, and this free oxygen will appear as bubbles. So appearing of bubbles from the colony indicate that this bacteria is catalase positive, while catalase negative will not produce the bubbles after addition of H2O2. The second biochemical reaction is the coagulase test. Coagulase is enzyme which coagulates citrated human or rabbit plasma. We have two types of coagulase tests. We have the slide method and the tube method. In the slide method, the coagulase enzyme will be bound to the bacteria, and it is done on the slide. The coagulase positive bacteria will produce white or macroscopic clumping around the bacteria, while coagulase negative bacteria, there is no clumps. This is the slide method. It is done after addition of rabbit or a human plasma to the bacteria on the slide and wait for 5 to 20 seconds. The second method is the tube method. It is done when the bacteria contain free coagulase. We will inoculate the bacteria in broth culture and then add the diluted plasma to it and incubate and examine the tube after different intervals. Now the coagulase positive bacteria will produce clots. As you see here, this is a clot formation. Is indicate that this bacteria is coagulase positive bacteria, while coagulase negative bacteria, there is no clot formation. Now Staphylococcus aureus is coagulase positive Staphylococci, while Staphylococcus epidermidis and Staphylococcus saprophyticus are coagulase negative Staphylococci. So if we examine a slide by the microscope, we will take the gram-positive cocci. This is for all Staphylococci. In order to differentiate it from Streptococci, we make the catalase test. If it is positive, it means that it is Staphylococci. Now, the catalase test will not differentiate between the species of Staphylococci. In order to differentiate between species, we will make the coagulase test. Staphylococcus aureus will be coagulase positive, while epidermidis and saprophyticus will be coagulase negative Staphylococci. The last point about the genus Staphylococcus is the cultural characteristics. As we said, the cultural characteristics include the growth requirements and the colony morphology. Now, Staphylococci can grow in different culture media under aerobic and facultative anaerobic condition. The colonies are circular, smooth, moist with an entire edge, and they can tolerate high concentration of NaCl. They can tolerate concentration of NaCl of 7.5 to 10%. And I will tell you the importance of this point later on. Now, regarding the cultural characteristics, the media where the Staphylococci can grow, we have the nutrient agar, the blood agar, and the mannitol salt agar, NSA. Now, on the nutrient agar, all Staphylococci will grow rapidly at 37 degrees centigrade for 24 hours. But the species will produce different colors of colonies on a nutrient agar, and they best produce this color on room temperature. It means we inoculate the bacteria and incubate it for 24 hours. After that, we leave the plates outside the incubator at room temperature, looking for the color of the... So regarding the nutrient agar, each species of Staphylococci will produce a characteristic color. Now Staphylococcus aureus will produce golden yellow colonies. As you see here, these are the golden yellow colonies produced by Staphylococcus aureus. Staphylococcus epidermidis will produce white color colonies, and Staphylococcus saprophyticus will produce lemon yellow colonies. The color is like this. This is a lemon color colonies produced by Staphylococcus saprophyticus. Now regarding the blood agar, the Staphylococcus aureus will produce beta hemolysis on blood agar. So as you see in this picture, there will be formation of a clear zone surrounding the colony, as you saw it previously. This is in Staphylococcus aureus. While Staphylococcus epidermidis and saprophyticus are non-hemolytic on blood agar. So there is no hemolysis surrounding the colony. As you see here, there is no clear zone around the colony. This is in case of Staphylococcus epidermidis and saprophyticus. Now regarding the mannitol salt agar, it contains beef extract, peptone, and contain the NaCl in high concentration, contain the mannitol, and also it contains the phenol red, which is pH indicator. pH indicator is a chemical substance. Its color change when the pH of the medium is changed. Now phenol red is normally red in color in neutral pH. But in acidic condition, when the pH decreases, its color will change from red to yellow. This is about the phenol red, which is the pH indicator. Now the mannitol salt agar is both selective and differential for genus Staphylococci. Now it is selective because it contains high concentration of NaCl. So only the Staphylococci will grow on this media because it can tolerate the high concentration of NaCl, as I mentioned previously. While other bacteria, since they cannot tolerate this high concentration of NaCl, so they will not grow on mannitol salt agar. So it is selective for genus Staphylococci. Also, this medium is differential by the presence of mannitol. It is differential for the species of genus Staphylococcus. According to the fermentation of mannitol, Staphylococci will be differentiated into mannitol fermenters and non-mannitol fermenters. Now, Staphylococcus aureus is mannitol fermenter. So it will ferment the mannitol, it will produce acid, and this acid will change the color of the phenol red from red to yellow. So as you see here, this is the Staphylococcus aureus. Due to the production of acid, it changed the color of the medium from red to yellow. This is in case of Staphylococcus aureus. While Staphylococcus epidermidis and saprophyticus, they will grow on the medium because they can't tolerate the high concentration of NaCl, but because they are non-lactose, non-mannitol fermenter, they will not produce acid, so the color of the medium will remain red in case of Staphylococcus epidermidis and Staphylococcus saprophyticus. So mannitol salt agar can differentiate between species of Staphylococci. So in summary, regarding the genus Staphylococcus, we talk about the classification. They are classified into pathogenic, Staphylococcus aureus, and non-pathogenic, like epidermidis and saprophyticus. Regarding the microscopical appearance, they appear as gram-positive cocci arranged in clusters. This is the microscopical appearance. The third point is the biochemical reactions. We have the catalase test. The Staphylococci are catalase positive, while Streptococci are catalase negative. All species of Staphylococci are catalase positive. Regarding the coagulase test, we have the slide method and tube method. Staphylococcus aureus are coagulase positive, while epidermidis and saprophyticus are coagulase negative. The last point is the cultural characteristics, which include the nutrient agar, blood agar, and mannitol salt agar. Now, on the nutrient agar, each species will produce a characteristic color. Aureus will produce golden yellow colonies, epidermidis will produce white colonies, and saprophyticus will produce lemon yellow colonies. Second is the blood agar. Staphylococcus aureus produce beta hemolysis on a blood agar clear zone, while epidermidis and saprophyticus are non-hemolytic on a blood agar. And 3 is the mannitol salt agar. Staphylococcus aureus will produce yellow zone surrounding the colonies because it is fermenter for mannitol, while epidermidis and saprophyticus will grow on mannitol salt agar but without formation of yellow zone surrounding the colonies. So this is all about our lab today, including the isolation of microorganisms from the environment and genus Staphylococcus. And thank you very much.